Welcome. Okay, so good morning, everybody, and welcome to the 2014 Lyell meeting. Oh, yeah. That it is, as you know, on chemosynthetic ecosystem this year. So my name is Silvia Danise. I'm a postdoc researcher in paleontology at Plymouth University. And together with Chris Little, who is a senior lecturer at the University of Leeds, we had the great pleasure to propose and organize this year's event. So I cannot start without saying thank you to the Geological Society for hosting us today, and also for the great organization and support for today's event. And also I need to thank all the other members of the Joint Committee for Paleontology that makes this Lyell meeting event possible every year. So this is the Micropaleontological Society, the Paleontological Association, and the Paleontographical Society. And so this year, we also have been sponsored by the Challenger Society for Marine Sciences. So, I mean, our acknowledgement to them as well to sponsor us. So why this meeting today, why it is important? Chemosynthetic ecosystem and their discovery has been considered like one of the major highlights on the 20th and of the 20th centuries. And this is across all the field of scientific research. And they were discovered in the 1970s of the past century. And why they were so important? Because they completely changed our idea of the deep sea. So the discovery of this kind of abundant organisms associated with hydrothermal vent system, they were rich in biomass and highly biodiverse, really changed the idea of a deep sea that was barren of life and barren of primary productivity because of the absence of sunlight. This was a really big change in our idea of how deep sea systems are. And then, knowledge goes very, very quick in this field. So what we know now it is that chemosynthetic ecosystem that don't just occur at hydrothermal vent system, but this kind of unique system that can live even in other extreme environment, such as the cold seeps and large organic falls like whale falls. And then we know even more also on their evolution and the fossil records. So here you've got Chris Little, if you don't know him personally, that is pointing on some limestone Miocene cold seeps from New Zealand, and here you've got a fossil whale from the Pliocene of Italy. So why we decided to do this meeting? So basically it was kind of to celebrate these 40 years of great discovery and exploration in the deep sea. And so we decided to bring together the kind of main researcher in the field from different disciplines to tell us, to summarize what is our current knowledge of chemosynthetic ecosystem, of their history, how they work, and what are the unique adaptations that they have. So it is kind of really evolving environment. So what I did, it was to do a small research on the Web of Science to see how much it was published since the discovery of chemosynthetic ecosystem. And what you have here is that, incredibly, more than 80,000 papers have been published, and these are the keywords that I use. So there's a small peak here for the 77 when they were discovered, and then something here in 84 where cold seeps were discovered, and then you can see there is a big increase, a number of paper increase every year. But also what I noticed, so if you split these in kind of disciplines, so what has been published and who are the people involved in this research, it is really interesting because it is really, for me this is why it's so amazing field, because it's really multidisciplinary and interdisciplinary. So you've got geochemistry and geophysics that of course you need if you want to understand the fluids that are governing these systems in hydrocarbon and hydrothermal vents. And then you've got marine biology, oceanography, microbiology, and all other disciplines of biology. Geology is very important, of course, together with paleontology, and we also got publication from astronomy and astrophysics, and of course astrobiology. This is the related discipline to it. But the last thing that I want to point out is that chemosynthetic ecosystems in the deep sea, they are not just important because of their incredible biodiversity and for their unique adaptations, and this is what we discovered today, but they are very important for the future and for our resources. So here below you've got other things. So they are important for mining, they are important for energy fuels, and so they need tools from engineering ocean. So our resources are kind of decreasing in shallow and on land. So industry is looking for resources in the deep sea, like deep sea mining and working at hydrocarbon seeps and methane hydrate, looking for them. And so it is exactly 1 year ago, on the 12th of March 2013, that there was published the news that Japan was the first to extract gas from methane hydrate. So we don't know which are the implications for the biodiversity of these systems, but we can probably start what we know. So we can start with our meeting and see what we know, and then let's see in the future what will happen with the exploitation of these resources. So I would just thank you, all of you, for being here, and we can start with our first speaker, which is, yes, Jonathan Copley. And okay, his talk today, it is, we have a little bit of Latin here, so Orbis not sufficit, going beyond biogeography in understanding ecological variation at hydrothermal vents.
Thank you, Sylvie. Good morning, everyone. What I'm going to talk about today is very much part of a much bigger collective effort than just me as the sort of named author, if you like, for this speech. So I would like to thank the many students that have been involved in my group, who've all contributed to the work I'm going to be talking about, and international colleagues, a great list you can see there, who also have contributed to what I'd like to tell you about. And inevitably, with this field of research, the many wider shipmates that we've had on our research cruises in recent years all over the world. I'd like to start, though, with some history. And the first image of modern hydrothermal vent fauna that we now recognise was actually taken in 1966. And it was taken at 60 degrees south in the Southern Ocean. Now, in 2009, the NERC-CHESO Research Consortium, we had a research cruise to the East Scotia Ridge, and we pinpointed hydrothermal vents. We knew there were sources there 10 years previously. We pinpointed vent sources, the vent fields on the seabed, and got pictures of them with a camera sled. And at a project meeting after that first research cruise, we showed those images. Katrin Linzer, who's here today, said, hang on a moment, I've seen that marine life before, and she went off and dug out this monograph on Antarctic barnacles, published in 1971, with 2 seafloor photographs in it from this location on the East Scotia Ridge, which had this surprising abundance of fauna for the depth of 2,400 metres. And perhaps even more remarkably, this surprising for the deep Southern Ocean occurrence of this Anamuran crustacean, this crab, which we now recognise to be a new species of kiwa, which is particularly abundant, as we'll see later, at the nearby, very close by, high temperature black smoker hydrothermal vents, which weren't photographed. So we've got the vent fauna here, but not actual venting observed on the seafloor. And it went unrecognised until we were able to go back and show that there were vents here, and this was the marine life associated with vents in this location. So, of course, if we think about the actual visual confirmation of hydrothermal vent fields on the seafloor, as we heard from Sylvia, then late 1970s, Galapagos Rift, Cameratoe in 76, dives in Alvin there in 77, actually seeing the venting on the seafloor. And now, if we have a look at where are we, how many vent fields worldwide have been visually confirmed on the seafloor, it is more than 200. And there's the sort of discovery curve, if you like, for that. And in addition, we know that there are at least 300 locations where there are water column signals indicative of underlying hydrothermal activity, but where that source hasn't been pinpointed and visually confirmed yet on the seafloor. And of course, some of the sort of landmarks along that road, the first high temperature black smoke events, on the East Pacific Rise in 79, Northeast Pacific Ridge System shortly after that, then the Mid-Atlantic in the mid-80s, then Western Pacific around about the same time, a year later, and then quite a gap until people got into the Indian Ocean, just at the start of the millennium, Arctic vents in the middle of the last decade, and then we saw those high-temperature Southern Ocean vents for the first time in 2009. So there's our sort of discovery curve for visually confirmed vent fields. If we have a think now about new animal species that have been described from vent environments during that period, well, here's what the collector's curve looks like for that. And so it passed 400 about 7 or 8 years ago. And ones you're probably familiar with, okay, we've got, the cybergliding polychaetes, riftiopachyptilla, that was a public description published in 81. We've got, the alvinicaridid shrimp Rimicaris was 86. We've got the snowboarding scaleworm, Bathycorilla guimonensis, and of course this is one that yes, found and described from vents, but also we now know occurs in other chemosynthetic environments in the deep ocean. And more recently, we've got, the first of the species of kiwa, the yeti crab, Kiwa hirsuta, more recently. Okay, so if you look at these two curves, you might think, well, on average, if you rock up at a new vent field, then on average you might expect to find 2 new species. But of course it doesn't work like that. And it doesn't work like that because these species are distributed in biogeographic provinces. So we will have a region where the vent fields share species among vent sites across that region, and then it will be different species among vents in a different region, and often some sort of geological or oceanographic barrier between the regions. And this has been one of 2 key themes, I think, in ecological research at hydrothermal vents, filling in the gaps in our global biogeographic map of vent fauna. And so this is a map that was published in a paper in 2010, and you can see it actually highlights missing jigsaw puzzle pieces. And so this has been a key driver in a lot of our research, going to new areas, looking to see, do they have distinct faunas, what are the relationships with faunas of different regions, and so on. At the same time, the other, I think, theme in vent ecology has been carrying out studies at sites that we've already found in established provinces to look at their temporal dynamics and compare them between provinces, and to carry out, often experimental work to look at process studies involved in what's structuring the sort of assemblages and driving the dynamics that we see. So we've got these 2 themes, but biogeography has been certainly a main driver in one aspect of the work that we've done. So what I'd like to talk to you about this morning then is basically an update on vent biogeography. Now we have several of the missing puzzle pieces. So what's in those missing puzzle pieces, and what does the new expanded global picture show us? And in particular, now we have that new expanded global picture, can we use it to perceive ubiquitous ecological patterns at hydrothermal vents? And secondly, what doesn't it allow us to perceive if we're just looking at biogeographic patterns? What's the nature of the ecological variation beyond species, presence, absence biogeographies? And that one is going to be particularly important because it's that kind of appreciation and understanding variation we're going to need for the management of human activities in these environments in the future. So that's where we're going to go this morning. So I'll start with a quick update on vent biogeography. There have been quite a few biogeographic province schemes published and refined and developed over the years as our knowledge has increased. So you can see one here from 2002, a paper in Science. There's a more recent one that's specifically vent biogeography paper from 2009. There are lots of different ways you can do this. In some cases, it can be quite hard to get the details of the method that's generated a particular scheme, and in some other cases, we now know that there are some issues with some of the details of the methodologies that generate these schemes, but largely they show very similar pictures. But, however, even some of the most recent ones, here's one from 2010, they don't incorporate the results from the most recent exploration of new areas that's been going on over the past 5 years or so. So that's what I'd like us to have a look at first this morning. And I'll share some of the work that my group has been involved in. We've been involved in this UK consortium looking at vents on the East Scotia Ridge down in the Southern Ocean. We'll also, though, I think, have a look at the other end of the planet and work that Norwegian colleagues have been doing at vents north of Iceland, getting up into the Arctic Ocean, and the fauna being revealed up there as well. Then we've also been working with a lot of international colleagues on the Mid-Cayman Spreading Centre in the Caribbean, so we'll have a look at that. We've also, with Irish colleagues, been to the first deep water vent field on the Mid-Atlantic ridge north of the Azores, and we'll see some results from there. And finally, one of the big gaps on the map, the Southwest Indian Ridge, ultra-slow spreading Southwest Indian Ridge, and I'll show you some preliminary results from there. And then we'll put it all together and see where that takes us. So let's start off then down south in the Southern Ocean, and this is work that the sort of discovery paper, if you like, was published back in 2012. And what have we got in terms of fauna on the East Scotia Ridge? Well, at species level, it's distinct from everywhere else. So in a sense, that allows us to propose it, erect it as a new province of vent biogeography, if we take that as our definition. What are some of the key abundant fauna that we can see when we visit these vents? Close to the high temperature vent sources, we have these amazing abundances of this new species of kiwa crab. Okay, manuscript name at the moment, Kiwa tyleri. Hopefully that'll be published very soon. And so, yeah, amazing abundances, as you can see here, just absolutely stacks of them. And of course, previously, Kiwa hirsuta from Pacific vents, relatively low abundance, much more peripheral in its distribution. So some interesting ecological differences and morphological differences within the genus. Gastropods as well, abundant in these systems, and there's a very large peltospiroid you can see here. This is work led by a PhD student I co-supervised, Chong Chen, at Oxford, and a new genus, Gigantopelta here, Chesoia, and that's going through review at the moment. And there's also Lepidodrillis, and I'm sure Captain Linzer can tell you much more about the Lepidodrillis if you want to find out more afterwards. Moving further out, if you like, from the high temperature vent sources, then there's a new species of eolepidid barnacle that's been published now, Volcanolepis scotiensis. There's also a couple of species of actinostolid anemones, which are very exciting. And then when we get into the periphery, one of the, to me, notable organisms in the periphery is this seven-armed stochastrid sea star, which is a very, very lovely animal to see. And in fact, when we were coming up on the vent fields with the tow camera in 2009, it was starting to see an increasing abundance of these after many, many hours of going over the seafloor that gave us the clue that we were getting close. That's what triggered the excitement. Okay, so the Southern Ocean, the fauna, species level distinct. Some of the interesting features to note. What we have here in particular is, as you might expect, at these latitudes, we have an absence of taxa with planktotrophic larvae, okay? So we don't see the Bathymod Island mussels, we don't see the albinocaridid shrimp that often dominate other systems. Let's go to the other end of the planet. Let's go into the Arctic, and this is work by Norwegian colleagues. So here we're talking about the Moan Ridge, north of Iceland, and the junction of Moan Ridge and the Nipovich Ridge, which is where this site is too. Some sets of vent fields there. Some of them are relatively shallow, 500 to 750 meters, and then Loki's Castle, 2,400, and there are further discoveries more recently up in this area, gradually pushing further north. So, okay, fauna here. The usual suspects from the northern mid-Atlantic ridge south of the Azores, not present here, So, and again, some big differences between shallow and deep and so on. The deep vent's very interesting. There's this melitid amphipod that you can see here, which is lovely. And affinities here, depending on how you do the analysis, there's Nicomachi, there's a polychaete, which is known from the northeast Pacific. Vent systems there, but then again, it's really, perhaps it's not, polychaetologists can tell us more about this, no doubt, but perhaps it's more of an organic enrichment sort of opportunist as a taxon. But some affinities with shallower seep-type environments in this location, certainly, but clearly big difference from vents that we know of south of Iceland. And again, if we look at it, we can see filters on certain features of life history biology. And again, no surprise, not that long ago, this area was covered by an ice sheet hundreds of meters thick. That will have effectively switched off surface photosynthetic primary production in this ocean. So when that happened, your deep sea fauna, relying on ultimately phytodetrital origin material, will have been pretty devastated. Chemosynthetic fauna may have been the only inhabitants of the ocean at that time, and those chemosynthetic, insular systems, well, planktotrophic species with planktotrophic modes of larval development for dispersal will also have been filtered out because there won't have been any phytodetrital material to feed during that planktotrophic dispersal. So again, we can see these sorts of features at the other end of the planet as well. Let's go somewhere a little bit warmer, the Mid-Cayman Spreading Centre in the Caribbean Sea. So what we have here is a 110-kilometre ultra-slow spreading, ultra-slow seafloor spreading centre. And it runs north-south across the feature, sort of generally known as the Cayman Trough. So that the trough is east to west feature, and then the Mid-Cayman Spreading Centre, 110 kilometres north to south across it. It's an extremely deep spreading centre, so I mean, it approaches 7,000 metres at the ends, and two active high-temperature hydrothermal fields, now recognised and studied, visited quite a few times now in recent years with us and international colleagues in this location. So we have the Beebe vent field, which is actually the world's deepest known vent field so far, just short of 5,000 metres deep. And that's kind of where you might expect to find a vent field. It's associated with a neovolcanic ridge, pretty much at the heart of the spreading centre. And I'll say a little bit about the fauna in a minute. But we also have the Von Dam vent field, which 2,300 metres on the upper slopes of an off-axis oceanic core complex. So a really different geological setting, and one where you might not expect to find high temperature activity. And the key point about this is not like Lost City on the mid-Atlantic ridge. This is high temperature, acidic, sulfidic venting. Okay, this is not a low temperature alkaline system like Lost City. Consequently, it has a rich, abundant associated chemosynthetic fauna. Different to Lost City as well. Lost City is quite a bit shallower, which is another filter, but very interesting system. You wouldn't necessarily have gone looking that far off axis for an active system, and to be honest, until recently, the traditional sorts of techniques for plume surveying and so on, you probably wouldn't have detected it because the particle load in the plume is very low here. It's quite hard to find. You have to use EH sensors, and we used AUVs and a whole bunch of other stuff. But it raises the question, maybe 50% of slow and ultra-slow spreading segments have these oceanic core complex features. If there is a common process that can drive high temperature venting there, how many of these are out there that we don't know about in those settings? Does this actually answer some questions about connectivity in systems on slow ridges? Who knows? So I think there's, it raises some interesting possibilities elsewhere. If this isn't just a sort of one-off on the Mid-Cayman Spreading Centre, there's only one way to find out, of course. So in terms of fauna, well, okay, there's a very abundant alvinicaridid shrimp at both sites, which I'll talk about in a moment. But so far, the species we've looked at, with only a couple of exceptions at the shallow site, are new, are distinct from, but closely related to faunas of other regions. So you can see, actinostolid anemone here. We've got alvinicaridid shrimp, I'll say a bit about. We've got this zoarcid fish, it's a pachycara, which we're working up at the moment. It's a new pachycara. But quite close to thermophyllum from the mid-Atlantic ridge so far anyway. So, okay, there are some interesting associations. So we'll have a look at those in more detail. So three species that one of my PhD students, Verity Nye, has led the description of. First of all, the only species so far that is common to both vent fields is the alvinicaridid shrimp, okay, Rimicaris hybisae. That's interesting because the two sites, they are only 20 kilometres apart. It's a largely isothermal water column between them. There's no sort of major oceanographic boundary between them. And yet the only shared taxon we've found so far is this shrimp between the two sites. Geochemically, they are quite distinct in terms of environments, which is, I think, very interesting. And there are clear phenotypic differences in the shrimp populations at the two sites. The one at the deeper site, the shrimp tend to be a bit smaller. And certainly when we look at their microbiota and so on, that's where it gets interesting because of the differences in geochemical environment. But okay, Rimicaris hybisae, so, in the same genus as Rimicaris exoculata from the Mid-Atlantic ridge, Rimicaris cairae from the Indian ridges, and pretty darn close to Coracaris chasei, morphologically and in other ways, and it's a safe bet that Coracaris chasei will soon revert to being Rimicaris chasei again, because it's also quite different to the other Coracaris as well. So there's some sorting out to be done in the group there. Now, at the shallower Von Dam vent field, we have another shrimp, but this is a hypolitid shrimp. Okay, so this is a different group of shrimp, and this is the first hypolitid at vents, in vent environments outside of the Pacific. So there's an interesting, evolutionary link, historical link. Of course, we've got a lot of gaps elsewhere, of course. There's also this lovely gastropod as well, and again, this is within a group where there's some sorting out to be done, but that's quite abundant at the Von Dam vent field too. Okay, and so the fauna is still emerging, and Glover is very involved in the polychaetes, and we're gradually working it up and getting a fuller picture. Okay, let's go to the Mid-Atlantic Ridge, though. It's north of the Azores. So this is the first vent field in deep water on the Mid-Atlantic Ridge north of the Azores, so between the Azores and Iceland. An interesting place to go, because the question is, does the shallow Azores plateau represent some sort of biogeographic barrier? Are we going to see a different province? So there were indications of vent plumes at one location during some geological work in 2007. We managed to get back there working with colleagues in Ireland, Irish Research Ship and Ireland's Holland 1 ROV in summer of 2011. And it was a very interesting trip. We were pretty confident that the venting would be on a near volcanic ridge in the, on the axial valley floor at a maximum 2,800 meters. Turns out the vents are in quite an unexpected setting. They're right over on the foot of the axial valley wall and at 3,000 meters. And we had that coincidentally was the depth rating of the ROV we had as well. So we were right at the limit of the ROV's capabilities. We certainly got to see the tops of the chimneys very thoroughly, and we did get to see the base of the chimneys, but below the base of the chimneys that we visited was out of reach to us. So there is some further exploration possibly to be done. But in terms of the fauna we found and were able to sample, what did we find? It is largely the usual suspects that we know from the vent fields on the Mid-Atlantic Ridge south of the Azores. So we've got Peltospira smaragdina up there, Myricaris fortunata, Protolyra valvatoides, we've got Pachycara here, we've got a Seconzacea, and there's some interesting things emerging from that, but largely the usual suspects, okay, in this location. But as we'll see later, the relative abundances do vary. Okay, so it's when you visit the site, it doesn't feel entirely similar to other sites we're familiar with. But that's actually, I think, keyed us into something else on the Mid-Atlantic Ridge, which I'll talk about at the end. Okay, let's now go to the final gap I wanted to briefly have a look at, the Southwest Indian Ridge, so the ultra-slow spreading Southwest Indian Ridge. So vent fields seen on the Central Indian Ridge, intermediate spreading north of the Rodriguez Triple Junction. Here we are, running southwest of the Rodriguez Triple Junction, and it's ultra-slow spreading here. So this is a vent field. Several vent plumes have been detected along the Southwest Indian Ridge decades ago now. And then in 2007, a Chinese expedition borrowed one of the AUVs from Woods Hole, and they got the first photographs locating the vent field on the seabed. But with an AUV, obviously not possible to collect samples and so on. So we were able to go back in November 2011. There was a UK research cruise looking at seamounts in the Indian Ocean with an ROV, and I was able to get some bolt-on time, divert the ship for 3 days to visit this vent field for the first time and collect the samples from it. So a very intense 3 days working with the ROV, but you can see some of the vent chimneys here. The Chinese colleagues had nicknamed it the Dragon Vent Field, so we kind of made that the theme for naming the individual chimneys and so on. But some spectacularly large sulfide edifices here, 20 meters high, a lot of extinct pinnacles, an extensive field, ultra-slow spreading, activity here, probably very prolonged looking at the size of the deposits, cycles of activity, all the kinds of things that we might expect in this setting. Okay, what about the fauna? Well, some real exciting surprises. So, let's go have a look at some of the fauna. There is a kiwa, yet another kiwa. It turns out it is distinct from, it's morphologically very, very similar to kiwa tyleri from the East Scotia Ridge, the Southern Ocean vents I showed you earlier. There are some differences, and certainly we can look at the CO1 divergence and so on. So it's close, 1.4 million years divergence, something like that perhaps. So there's a kiwa, but interestingly, compared with the Southern Ocean, where you saw that huge abundance, here it's comparatively sparse. It's in the same sort of location in zonation. It's close to the high temperature sources, but we've been diving for a while before we spotted them. So they're not there in the same abundance at all. There is another giant peltospiroid. This is another Gigantopelta, so that's the new genus that Chong has erected, and this is Aegis here from these vents. But again, in relation to East Scotia Ridge in terms of member of the same genus. There is also the famous scalyfoot gastropod, known from the Central Indian Ridge vents. That's present here. And so other taxa are shared with Central Indian Ridge vents. We've also got Miracaris here that's shared, a species that's shared with the Central Indian Ridge, and a bunch of other fauna as well. There's a Volcanolepis here, another eolepidid barnacle. There's a Bathymodiolus, it's the one we know from the Central Indian Ridge. There's also a Chirodotid holothurium, which has since been found, as Japanese colleagues have explored further north on the Central Indian Ridge. So that's also known up on the Central Indian Ridge. So we're seeing these affinities, but a bit of a crossroads, certainly with a Southern Ocean fauna and interesting possibilities with the circumpolar current down there and so on. And its history and its fluctuations may have deposited things here and then isolated them. So some interesting work to do there. Okay, what happens when we put all of this together? So we're in the process of working up a new sort of global biogeographic picture, and the method we've chosen to approach here is, well, for now, genus presence-absence data. Okay, so compiling that for known vent fields from the very helpful Vent Fauna Handbook, with which many of you will be familiar. You do have to do a bit of pruning of the Vent Fauna Handbook if you're going to use it for these sorts of analyses, though, because it includes, in some cases, records of peripheral fauna that were previously described from non-chemosynthetic environments. And sometimes when someone is doing a site description, they include the halo fauna, the non-vent, non-chemosynthetic obligate sort of taxa, which is great. But then some people don't do that in the description, but that's not a real difference biogeographically in terms of vent fauna. So we restrict ourselves just to the vent, obligate fauna, if you like. So we do a bit of pruning of the data set that people use for this. There are a few updates and corrections, and then, okay, we can code in all of the new areas, from published papers of other workers, from our own work. And then for this, we've chosen to use Sorensen's index as a simple, sort of measure of sort of shared genera. Single linkage, because that to me is more forgiving of differences in sampling effort. Where we recognize an affinity, we will take it at a certain level. Therefore, if more work has been done and we have a more expensive species list, that's less of a factor in the patterns that are generated. So there are some choices to be made there. And then running that through and doing this sort of classification, and then running a SimProf test can be quite useful. So SimProf allows us to say, where are the affinities? Where is there no more coherence, if you like, within a group, which to us means, that's actually a group of a set of sites with very shared taxa. So when we do this, we get this pattern here. Just to take you through a few features of it. So what I've done is color code things by region, but where we've got a SimProf group, I've bound them together with a line. Okay, we've got Western Pacific here, and we've also got a few Western Pacific here. We've got some closely located, well-connected sites and well-connected among each other, and we've got some that are in very isolated basins. The Western Pacific is a bit of a mixed bag. Indian Ocean and Mid-Atlantic are pretty close in this. All of the Indian Ocean and the Southwest Indian Ridge fits within the others at the moment. Mid-Atlantic deep and shallow we can separate off here, which is quite interesting at this level. They are separate in this. Cayman are distinct to each other, but come in here as well. Anyway, one of the things, we go to the East Pacific Ridge, East Pacific Rise, you'll notice it's pretty fragmented. Okay, why? Here we have high rates of vent field scale disturbance. We therefore have vent sites at different stages in succession, which therefore look distinct. Mid-Atlantic Ridge, long-lived, okay, with a lot of plankstroke larvae, panmictic populations, that kind of thing. We see, so with ridge spreading rate, we can now kind of look at the fragmentation through this kind of analysis, which allows us to go a little bit further. Okay, so moving on rapidly, ubiquitous ecological patterns. Zonation is definitely a big one at hydrothermal vents, of course. What's actually driving it? I'm sure we will hear more from Nadine about this presently. Can we come up with and succession is the same as zonation, but it's a pattern in time rather than space. Can we come up with a universal zonation scheme like we have for the intertidal? Based on animal taxonomy, it wouldn't seem that we can. The players are different in the different zones and different provinces. But when we think about these animals or the organism as a holobiont, and we look at the microbes with which they are associated, then that becomes key. And we can actually make a division based on metabolic flexibility and restriction between epsilon proteobacteria holobionts, gamma proteobacterial holobionts, and that may be quite key in driving our zonation. So we often get epsilon proteobacteria dominated holobionts close in. The identity of the animal may change, but that's the functional holobiont, if you like. Then we see the gammas further out because they have these metabolic, more restricted metabolic pathways in terms of demands for, particularly for a terminal electron acceptor. And then we see other trophic modes, suspension feeders, predation beyond that. So possibly a universal functional zonation effects. And just to finish off then, this is only part of the story. There is clearly key ecological variation within biogeographic provinces, and we see that perhaps most clearly on the mid-Atlantic ridge. So here are all the mid-Atlantic ridge vent fields north of the equator, and here I've done it so you can actually see ridge axis bathymetry and distance. And if we look at the taxa, some of the key taxa, we look at the differences in relative abundance, we find a lot of variation. So for example, Rimicaris, some places incredibly abundant, some places very rare. Maybe it's a depth distribution thing going on there when we look at that in more detail, but for other taxa, Miracaris, varies in a different pattern. The so Peltospira, which we saw at Moitera, yeah, again, very variable, not depth, not latitude, something else is driving that variation. Same goes for the Bathymedilans as well. We have a change in species along the ridge, but nevertheless, we see some differences there, and very much so in the actinostolid anemones as well. So there is a key variation that we do not capture through biogeography, and we're currently working on a new metric that will capture or summarize that kind of information for each vent field and allow us to have a quasi-quantitative measure of ecological distinctiveness among vent fields to then explore this further. And then we can code that for space. We can examine spatial structure through that kind of analysis. We can also look at perhaps functional traits and start to explore those with that kind of tool. So that's where we're headed next. Thank you very much. Yes. Oh. Yes, I had seen that.
Is it related to venting system?
I don't know. Yes, I had heard about that, and I don't know, and I'm not aware of those sorts of effects having been detected at any vents yet. I have to say, starfish are rare at vent. There aren't that many. This is one of the few examples. So I don't know if that's also operating worldwide here. I mean, who knows? Certainly something to keep an eye on. Hmm. So circulation, indeed, big questions, and that's. I don't know, but certainly one of the things why the Arctic is it's not just that history that's made the Arctic vent fauna different. It's also, of course, hydrography is currently exporting from the Arctic. It's hard for anything to get in there now since that's opened up again, or since primary production for photosynthetic food chains has been switched on again effectively in that ocean. So what further changes lie ahead? I don't know. I don't know.
Thank you for the questions. Thank you very much, John, for your talk. Okay, so